Okay, this will be a short recording on Mendel's law of independent assortment. You should watch this recording after the recording on Mendel's law of segregation. So for this section of genetics, you need to be able to describe the law of independent assortment and understand the difference between a monohybrid and a dihybrid cross. In the previous video, we did a Punnett square that had 4 squares. So this is a much more complicated Punnett square. You can see here it has 16 squares. You do not have to actually do a cross with 16 squares for this course. But what I do want you to understand is that this type of Punnett square is used for what we call a dihybrid cross, meaning that we are looking at the inheritance of 2 genes at the same time. Whereas monohybrid cross, mono means 1, you are looking at 1 gene. So let me write that down for you. So dihybrid, di means 2. You are looking at the inheritance pattern of 2 genes at the same time. There you go. So now we will talk about the law of independent assortment. So here we have chromosome pair number 1. And I'm going to put 2 genes on these chromosomes. So we will look at flower color. So we have a capital F and a lowercase f. So this is a heterozygous individual. And now we are going to look at seed shape, round and wrinkled. Let me write the genes up here. So the first gene is flower color, the second gene is seed shape. So for this individual plant that I showed you right here, it is a purple flower, round seed plant, because it is heterozygous for both genes. Now let's go back and look at DNA synthesis. Remember, this occurs during S phase of the cell cycle. And we now are going to have sister chromatids in addition to homologous chromosomes. So sister chromatids, remember, are identical. So we have a big F and we have a big F. Okay, so this should look familiar to you. Now remember that crossing over occurs during prophase 1 of meiosis, and this is where alleles are swapped. Now, if you look back to the original plant, on one chromosome you have a capital F, capital R. On the other chromosome you have lowercase. The law of independent assortment means that the capital F is not necessarily attached to the capital R, right? You can swap those alleles, and here's what I mean by that. So let's say we have crossing over here where I put my circle. That is going to result in this. So here we have my sister chromatids again. So we had crossing over, which will give me a capital F here. But now, instead of a capital F on that sister chromatid, I have a lowercase. Here I have a capital R and a capital R. Now over on my other set, I have a capital F now because those two alleles swapped places. And I have a lowercase r on the bottom because it did not switch places. So what I want you to notice here is now I have a unique combination of alleles. Instead of having capital F and capital R and lowercase f and lowercase r, now I have a recombined or recombinant chromosome. I have a lowercase f and an uppercase r. So that is a new combination. I have now an uppercase f and a lowercase r. So those are two recombinant strands of DNA. They have a unique combination of alleles. So what I'm showing you here is that crossing over can occur. That is what the law of independent assortment means, is that crossing over can occur between genes, right? So here I have the genes for flower color, here I have the gene for seed shape. Crossing over can occur between the location of flower color and the location of seed shape. So let's go back here. This is what Mendel came up with for the law of independent assortment. Each pair of alleles segregate means to separate, independently of the other pair of alleles during gamete formation. Gamete formation just means meiosis. So that's saying that the alleles for flower color can swap independent of the alleles for seed shape. I'm going to stop this video here. My next video will be on pedigrees and inherited diseases.